Okay, whenever you're ready.
Okay. Howdy, my name is Cassandra Fernandez and Alondra Curiel, and today we're going to present our biochemical test two. Introduction. The lysine and ornithine decarboxylase test are biochemical assays used to differentiate and identify gram-negative bacteria based on their ability to decarboxylate specific amino acids. These tests detect the enzyme decarboxylase, which removes the carboxyl group from amino acids such as lysine and ornithine, producing an alkaline end product. The resulting pH change is indicated by a color shift in the medium. A positive result indicates the presence of the enzyme and helps confirm the metabolic characteristics of the organism. Materials. For the equipment, we used an inoculating loop, an inoculating needle, a Bunsen burner, and for the cultures, we use Citrobacter frundi, Enterobacter cloacae, E. coli, Morganella morgani, Proteus mirabilis, P. aeruginosa, Salmonella triferium, and then for the medium, we used LIA and ornithine broth. For the lysine decarboxylase test, we obtain LIA slants using a sterile inoculating needle. We stab the butt of the medium and then streak the slant with the organism. Then we loosely cap the tube to allow gas exchange. Then we incubated the tubes for 18 to 24 hours. Then we observed color changes. Purple indicated a positive decarboxylase reaction, and yellow indicates a negative result. For the ornithine decarboxylase test, we obtained ornithine broth tubes, inoculated each tube with a loop full of the bacteria culture. Then we overlaid a small amount of sterile mineral oil to create an anaerobic environment. Then we incubated the tubes for 18 to 24 hours. And then the results, if it were purple, it was positive, and if it was yellow, it indicated negative reactions.
For our results for our lysine decarboxylase test, for estifimurium, we saw a positive lysine result, which was interpreted as a lysine decarboxylase present and a it's typical for the salmonella. For M. morgani, it was a positive result, which is kind of unusual but possible, weak LDC plus, and there was a no deamination. For unknown, this is also our gram negative. We got a negative result. The lysine decarboxylase is absent. For E. coli, we got a positive result. And that just means the lysine decarboxylase is present for LDC and positive for IDC. And this is typical for a Proteus. For our ornithine decarboxylase test for C. fernundi, we got a negative result, which just means there's no ornithine decarboxylation and the medium stayed acidic. For E. coli, we got a negative result, which just means acidic pH from glucose fermentation. There's no alkaline reversion. And for our unknown, we got a positive um result, and this just means that the ornithine decarboxylated to putrescine, and the alkaline pH is restored. In this lab, several biochemical tests were performed to identify bacterial species based on their enzymatic activities. The lysine and ornithine decarboxylase test provided valuable insight into each organism's metabolic capabilities, particularly within the Enterobacteriaceae family. Distinct color changes in the media allow differentiation between decarboxylase positive, deaminase positive, and negative reactions. Results showed that E. coli and Salmonella typhimurium were positive for lysine decarboxylase. Proteus mirabilis was lysine deaminase positive, and Citrobacter frundi was negative for ornithine decarboxylase. The unknown 294 tested lysine negative and ornithine positive, suggesting it belonged to the Enterobacter genus. Overall, this experiment demonstrated how biochemical testing is an essential tool for identifying and distinguishing closely related bacterial species. Thank you for listening to our biochemical part 2 lab video. Okay.